Cool. Great. Well, thank you everybody for coming today to listen to cancer-specific gene lists. This is a topic that personally I've been working on for about a decade, both internally at the Mayo Clinic and then also collaboratively with groups at the Cancer Genomics Consortium to standardize a protocol for making these lists in a way where we know that they're created in a way where they will be clinical grade and also curated in a way to keep up with current knowledge. So today we'll talk about why cancer-specific gene lists are helpful, talk about clinically relevant genes, what kind of inclusion criteria we employed in this process, different kinds of supporting information that we include in the gene list, as well as a significance level that we assign to genes based on their maximum potential impact on oncogenicity. We also have curation recommendations, some future directions for maintaining these gene lists and for providing community or for incorporating community feedback. And then I'll run through one case example of a complex case that really benefited from the use of a gene list. So back in 2015, our laboratory went live with cancer microarrays. So we went live with OncoScan. We also had a lot of heme microarrays coming through our laboratory. And we ran into this problem when we had a complex microarray. How would we know that we caught the important genes in our report based on different cancer contexts? And if we would get like a cholangiocarcinoma, for instance, that was highly complex, but we have the expertise in lab in-house on that day to help us with that? Or could we make some resources so that not everybody had to necessarily be the primary content expert in order to make sure and be certain that we called out all of the important regions and genes in that cancer type. So the gene list came about as like a necessary tool to do this job so that we could make sure that we brought all of those genes to the forefront of the report, because a lot of times when you have a complex genome, if you just list all the abnormalities, the report ends up feeling kind of difficult to understand what the importance is or what are the important findings in this assay. So we need to know which regions should be included in a report and which genes to call out. This is an example of an astrocytoma IDH mutant. It's a fairly complex genome. When we added a gene list on top of this, we were able to see some important genes or identify some important genes. You can see these green arrows are at normal copy number state. B allele frequency looks normal. A lot of times TP53 will see a copy neutral LOH in this tumor type, but we did not observe that in this sample. We did see a small gain that was disrupting NTRK1, a loss of 5P, including TERT, a homozygous deletion of CDKN2A and B, and a loss of P10. This is an extremely important finding with the CDKN2A and B homozygous deletion because this actually confers a CNS WHO grade 4 tumor. So this is important for defining the grade of the tumor in this individual. So when we were talking with the CGC about this, we knew that the Mayo group, we called ourselves GOAT, so Genomics of Oncology Annotation Team. We had a protocol, but how can we apply this more broadly and in a standardized way where we can really leverage the expertise from the CGC working groups? We had shared a couple of our gene lists with the CGC as a supplement to their annual meeting back in 2017, I believe. We shared our B-cell ALL and our T-cell ALL lists with that group at that time. Following that, the CGC working groups had an interest in creating their own gene lists, and the CGC Education Committee had a desire to standardize the process. So we started working on this back in about 2021, where we identified two of these working groups that had an appetite for creating their own gene list. So this was the breast cancer working group and the myeloid neoplasms. We felt like this was a really good fit because while we're standardizing this process, we could ensure that the process would work for both solid tumor entities as well as hematologic malignancies. And both of these cancer types are really well studied as well. So there's a lot of molecular data out there to support kind of using a gene list for these. So kind of the first thing we do is we create a candidate list. So we kind of have a broad net that we cast to collect genes. So we'll do a literature review of more prominent literature with some standardized prompts that we'll put into something like Google Scholar. We'll put genomic profile plus a cancer type or genetic profile plus a cancer type and try to get these larger studies that have incorporated lots of genetic data and genomic data surrounding these cancer types. We'll also look at NCCN and WHO guidance. And then we used both the Cosmic Cancer Gene Census and the Atlas of Genetics and Cytogenetics in oncology and hematology more as a QC to make sure that we caught all of the genes that were attributed to our cancer type in those resources. If we found additional genes, then we would run additional literature search, specifically looking at those genes. Once we had that candidate gene list, we entered into more of a peer review process where we looked at specific criteria for keeping genes in the list. So we wanted abnormalities involving this gene to be reported in at least 2 case reports, at least 1 case report with functional data, 1 case report in 1 cohort study, or 1 cohort study with substantial data. And these, we tried to make this as objective as we could, with the understanding that different tumor types, if they're more studied or less studied, may have slightly different criteria. And then genes within genomic regions of significance were not just automatically added because they were in that region, but we needed additional evidence to support oncogenicity. Some large characteristic deletions and duplications or gains may contain hundreds of genes. So this is, we need to be able to pinpoint genes that are of actual clinical consequence. And then we included genes with known germline alterations associated to predisposition to the specific cancer type. And then also genes with altered expression with evidence for oncogenicity within that cancer type. So that peer review process, we had more than 1 disease expert look at every gene from that candidate gene list and determine whether or not we would keep the gene in our gene list or kind of put it into what we call the parking lot, where it's more of an emerging gene of emerging significance, but we didn't feel like it had adequate evidence for now. So this is kind of a schematic of that process. So the first reviewer would evaluate a gene and make an assertion of, do we keep or do we toss this gene? And then the second reviewer would also evaluate. If there's a discrepancy in the evaluation, it was brought to a working group discussion. The myeloid group met every week for 30 minutes while this was actively going on, and it was really a cool process to see. They were able to move and work collaboratively very quickly. So it was really a cool process to be a part of. If the consensus was reached between multiple reviewers, then the gene review was complete, and we could move on with collecting the rest of the data. So along with this, we also came up with a way to stratify genes, which is kind of similar to, if you're familiar with the ClinGen gene disease clinical validity curation process. This followed a similar route where we wanted to identify the maximum possible clinical significance a variant may have in each gene, with the understanding that all genes can have VUS or benign variants. So, and this clinical significance could refer to any type of variant in that gene. It could be attributed to fusions or deletions, duplications, point mutations, any expression, all of the above. So in our clinical significance 1, which would be our highest clinical significance level, we included genes with alterations that are characteristic and or highly enriched in a tumor type. And some examples might be JAK2, which is characteristic for myeloproliferative neoplasms, or ERBB2, associated with amplification in breast cancer associated with diagnosis and therapeutic potential. Level 1 can also include genes present in professional guidelines, such as WT1 or NTRK3. Genes with alterations that are diagnostic for a tumor type. I thought this was a really good example, and I'm not going to have examples for every one of these categories along the line, but I wanted to kind of illustrate what this meant. So genes with alterations that are diagnostic for the tumor type, RARA would be included in here because we know that PML-RARA fusions are characteristic and diagnostic for acute promyelocytic leukemia. We have genes with germline variants associated with well-established germline predisposition. So for this, we included GATA2. Deficiency is autosomal dominant predisposition to early onset MDS and AML. And BRCA1 germline variants, we know increase risk for breast cancer. Also in this level are genes that are targetable by FDA-approved drugs, like FLT3 or PIK3CA for breast cancer, and then genes with implications for treatment according to current professional guidelines. And this would include something like ESR1. Under clinical significance 2, which would be the next tier down, we included genes with treatment implications that are under investigation. So they do not have approved treatment yet. This would include FGFR1 for breast cancer. Also genes with alterations that support the diagnosis of a tumor entity. And then this would include something like MYB. And so we know that MYB fusions are present in adenoid cystic carcinoma and are part of the triple negative breast cancer subtype. And then also in level 2, we have common variable partner for gene fusions, and this would include something like PML, where RARA is the actionable fusion partner and can be seen with other genes beside PML. PML is the common fusion partner, so this would be included as a significant level 2. And then also the myeloid group found that they could stratify this significance level 2 further by the frequency of alterations in the gene of interest, and they found that very helpful. So CUX1 is a tumor suppressor gene that is commonly deleted in myeloid neoplasms. So this was a high frequency level 2, whereas CREBBP is rarely translocated in AML with CAT6A. And then clinical significance level 3 was reserved for limited or emerging evidence supporting association with a cancer type of interest. And so for myeloid neoplasms, CRLF2 was included in the list for level 3. Fusion is frequent in B-ALL, but there has been a case reported in AML and MDS. So the group felt like that was appropriate to keep it in the list. And for breast cancer, CBFB is observed in approximately 4% of breast cancers associated with a hormone receptor positive, HER2 negative lobular subtype. And then based on peer review, some of these genes that were clinical significance 3, as I was kind of mentioning before, these were removed from the list and placed in a parking lot for more of a focused future review. So if we do see something that is emerging and in the literature, then we can focus in on those genes and just make sure that and capture any literature that comes out on those genes in this particular disease context in the near future. So we did collect a lot of different information. My apologies for this. And some of this information included the Hugo gene symbol, genomic coordinates with a genome build specified, of course. For our purposes in this project, we collected information and genomic coordinates in both HG19 and HG38, both chromosome and cytoband for those who are doing chromosomal microarray or cytogenetic studies. This is important. The types of genomic alterations, which might include like a gene fusion, structural variant, copy number deletion or gain, as well as single nucleotide variants or expression that would fit into the types of genomic alterations. Functional mechanism could be gain of function, loss of function, both of those or other. Diagnostic significance was also included, and this would include things like things that are specific to a subtype of that cancer. So, for instance, if a gene were only mutated in triple negative breast cancer, that would be an important piece of information to keep in the cancer-specific gene list, and it could potentially help in diagnosis of a patient. The clinical significance level, which we were through, germline predisposition, if any, variant in that gene predisposed to cancer. We added references, which could be PubMed IDs, it could be WHO references, and then also any kind of comments and notes that speak to nuances within that gene were added kind of manually ad hoc by the reviewers. So here's an example of all of the gene information that was collected for ASXL1 by the myeloid neoplasms group. So ASXL1 is on chromosome 20, cytoband Q11.21. Here are the genomic coordinates for HG19 and HG38. This can be deleted or it can be an oncogenic sequence alteration that would be loss of function. It's seen in a number of different myeloid neoplasms that are listed, that are abbreviated here. It's clinical significance level 1. There is no evidence of germline predisposition. Here are the references, and here's the comment that was added by the reviewers, and which lists that the prevalence of this alteration and that it's associated with poor prognosis across the spectrum of myeloid malignancies. And it also can be associated with CHIP. So when the gene list was completed, we have a couple different formats that this is organized into. So we have the Excel spreadsheet that has the full information, full comment, everything. Also, we've provided bed files and AED files for the CGC gene lists. The bed files are really easy to embed into different workflows and visualization. And the AED file is specific for kind of the Thermo Fisher, previously Affymetrix microarrays. And the myeloid and breast cancer gene list will be available on the CGC website and as supplemental data to the pending manuscript that is now sitting with the board of directors at CGC and will be submitted to Cancer Genetics. So lastly, I just wanted to walk through a case demo. We have a 7-year-old male, so a pediatric patient with pancytopenia and acute myeloid leukemia. By chromosomes, this is a very complex karyotype with a number of different alterations going on here. Chromosomes and FISH were performed in an outside laboratory, so we kind of take that caveat to heart here. There's clearly a lot going on with this genome, so we need to figure out, is any of this worth reporting? So I'm just going to go chromosome by chromosome. What we saw, what we highlighted thanks to the gene list, and kind of what we decided to include in the report. So on chromosome 10, we have a number of different deletions. Most of these had no genes in the gene list. However, this deletion has a gene that is included in the gene list, this ANKRD26 in the region. If we look at the gene summary in the spreadsheet, all of this refers to germline variants and not necessarily to copy number deletions. And based on the size of that deletion on the previous slide, I highly doubt that this would be germline. And also with the level of deletion across here, this would likely, this would not be compatible with life if this were germline. So based on this, we decided not to add this gene to the report as an interpretation. However, if this patient did have sequencing performed and did find a mutation in here, then the overlapping deletion could explain kind of an added loss of function. But we can't call it a germline variant here. When we look at chromosome 11, moving on here, we see a number of different amplifications on 11q. So if you look down here, these are very high copy number states. Most of these have no genes on our gene list, but this one actually has an amplified KMT2A, also known as MLL. If we look at our gene list, we know that recurrent translocations happen in MLL, but also KMT2A amplification can be observed as double minutes. And if we go back to our initial case, let me go back here. We did see double minutes in this case. So my guess is that these double minutes, if we were to do a fish probe on this case for KMT2A, these would probably light up and be consistent with that amplification of KMT2A. So move us back forward here. And so this is seen in AML. AML cases with a complex karyotype, which is very consistent with this patient with myelodysplastic related changes. So we would include that in the interpretation, absolutely. And then also we had a number of alterations on chromosome 12. If we look here, there are some deletions that have no genes on the gene list, but we do have several deletions that contain genes that are on our myeloid gene list in these regions. So we look into all of these genes, and we would include ETV6 in our report because deletions and loss of function are included in complex karyotypes in pediatric AML. And these are associated with poor prognosis in AML. CDKN1B deletions are reported in, oh man, I forget what that stands for, but they're reported in different myeloid neoplasms, including complex karyotype AML. So we would include CDKN1B. AEBP2, although it is a tumor suppressor, it's not specific for complex karyotype AML, and we don't know that this patient ever had PV. So we actually did not include this gene on our final report. KRAS, we did include. Deletions may indicate a loss of normal copy number if a mutation is present. And then SH2B3, this is a tumor suppressor. However, we ended up not including this on the report based on kind of director discretion, thinking that this was not specific to our patient's entity. And then PTPN11 is a common tumor suppressor associated with poor prognosis and seen in pediatric tumors, so this was included in the report, even though it is a clinical significance level 3. Moving on to chromosome 15. We had one large deletion with no genes in our list. And then chromosome 17, we had two large deletions encompassing some major tumor suppressors. And we know that loss of TP53 is correlated with a complex karyotype AML, as well as loss of NF1 and SUS12. And so when we look at our gene list, our gene summaries, TP53 is seen in complex karyotype AML, and they're associated with an unfavorable outcome. SUS12 is also a common tumor suppressor and commonly co-deleted with NF1 and has been, reported consistently in AML. And then NF1 has been seen as a deletion, and is also has been seen in pediatric patients. So these were all included in the report. Moving on to chromosome 19, we had another large deletion. No genes in our list, so we didn't have to do anything with this case. Or do anything with that chromosome. Chromosome 20, we did have one deletion on chromosome 20 that had these two genes, this L3MBTL1 and SGK2. When we look at these gene summaries, these were helpful. However, all those these genes are on our list. There's no diagnostic, prognostic, or predictive information or specific associations with pediatric AML that would be helpful. So in this case, they were left off of the report. And so the case summary is right here. So we have this long list of alterations that are input into the report as a table. So you can see that this is very complex. We'll also have our complex nomenclature input into our nomenclature section. But I wanted to just copy a few paragraphs that highlighted kind of the utility of this gene list in highlighting these genes. So if this patient does end up getting sequencing done, their clinician will likely have these genes right in front of them, so they can see what is consistent with what was observed by microarray. But the main feature here is an AML complex karyotype and TP53 gene deletion are both considered unfavorable prognostic findings by NCCN. And then we always add clinical and pathologic correlation is recommended. So that is the case summary. And with that, I'd like to acknowledge all of the people, both the Mayo Goat and then everybody at CGC who has worked really hard to get this protocol together and to come up with a peer review process for these gene lists. So with that, I'll take any questions. Hi, Peter.
Hi, Beth. It's nice to see you.
Good to
I
see
haven't
you
been on
too.
in a while. I, I'm not even sure whether or not this is relevant, but I'm going to bring it up, okay?
Yep.
About 5 years ago, we published an article with a fairly large number of validated splicing mutations, 341,000 of them, okay? And we're in the process of converting them for ClinVar. It'll be the largest entry in ClinVar as far as I'm aware. So these are
Mm-hmm.
validated with expression data. And one of the problems that we have encountered is just within the last 5 years, gene names changing. And that's why I kind of tuned in today. Now, it seems to me that you start out with a premise that these have to be in your study that the genes have to be very well established before they can even be looked at on your list.
Yep.
But coming at this from a classical clinical genetics point of view, we all know that there are genes that are modifiers and they can affect drug effectiveness, they can affect severity, they can affect, diagnostic relevance, and they're kind of being left out by your consensus selection process. And the question I have is, if you were to dig a little deeper, I realize you've got more genes than you can handle already, but if you were to dig a little deeper and start to look at genes that might be less well established in the literature as having a modifier effect, but nevertheless having such, especially when you bring up these large regions of copy number gain and loss with nothing in them. That you may run into some of the same issues we are. Uh, some of the tools that we're used to using. So we've been using the HGVS software Python modules for converting our G dot notation into C dot notation so that we know that what we're looking at is inside of an express transcript. Um, and we're coming up with a lot of inconsistencies between different databases in how genes are named. And the question I guess I have is, have you also seen this effect in your work?
Yeah, we've seen genes, even I would say in the heme um, kind of field, a lot of people still use MLL for KMT2A and similar genes. So in our Excel document, um, we have aliases in there as well. Um, but we try to keep current with what the current Hugo name is, and then just to update that as needed. We do encourage updates on a yearly basis or when NCCN or WHO or similar guidance is updated. So hopefully we'd be catching those in somewhat of a timely manner. But yeah, I think gene names are across the board with databasing and kind of keeping a knowledge base of things can be problematic. I don't know if that answers your question. We try to
Well,
stay on top of it, but it is
we've
manual.
tried a variety of things, and believe it or not, NCBI Entrez gets us the most results
Yeah.
of all of, Ensemble Biomart, MyGene, um, NCBI Entrez seems to capture the most results. But we sometimes have, when trying to find a transcript associated, when we have a transcript that can be associated with more than one gene, two genes might not share the same transcript. I mean, it's really a bit of a mess. And the question is, how much do you lose because of that kind of a mess, right? In other words, when you find nothing in a region that's 10 megabases long, is it possibly because the gene name isn't been established very well?
Um, I'm not sure it's because the gene name hasn't been established. It's probably because the clinical significance of that region hasn't been established. And in a complex genome, we understand that not every single 10 megabase block is going to have, a gene that's going to rise to the top of a report. And I think that's okay, so long as we get those big hitters that are in guidelines and in prominent literature. Um, if we do see a gene is um, that is called something else, we would probably, you know, make a point to catch that. I see that there are a couple comments in our chat right now. Thank you, Wes and Aiden, for including those. So Wes says the VICC does support a gene normalizer tool that helps with gene names. So that could be something to look into. I know we didn't use that in this process, given that it's more of a manual curation that was done by our breast cancer working groups and our myeloid neoplasm working groups at CGC. I see Dr. Catherine Geyersbach is on here. She was leading the breast cancer group. And then also Aiden says that the HGNC helper is useful if you use R. So I don't know if you're using R, Peter, but that could help as well. And so those links to those tools and literature are in the chat.
Okay, thank you.
Yeah. Yeah. And I will also say that DGC Genomic Resources Development Committee is working to create a mechanism where the community at large can um, offer suggestions for updates to these gene lists as well. So if you are using a gene list and notice a gap or feel like there is a gene that should be added. There will be a form available at some point for you to kind of influence change in those updates. Additionally, there is a plan to have a form for requests for additional gene lists. And so if we have a group of people who would like to make a disease-specific gene list that does not exist, we can provide tools and resources for those groups to get started.
Hi, Beth. Hey, Beth, can I ask you a question? This is Obi. Oh,
hey.
It's a quick one. I haven't been to the CDC website that covers gene lists recently. Can you update us on how they're being disseminated? Like, I know previously there was like a bed files or something like that were being distributed. Is that still the mechanism, or is there something maybe a little bit more sophisticated now?
I think right now there's an Excel sheet that would have like a spreadsheet with all of the information, a bed file, and an AED file. But I think the team is open to suggestions if you would want something in a different format. I think the primary use of these gene lists initially was microarray and more of these like large genomic tests. So a bed file was easy to kind of integrate, and the AED file would fit into the microarray software. So that was kind of the initial thought process there, but we could absolutely explore other opportunities. I don't think it would be hard to put this into a different format.
Yeah, it might be worth thinking about just like a simple database or something so you could track versions and corrections to them more easily.
Yeah. Yeah, that'd be great. I mean, we could even ask like the CCGA, which is also run by CGC, if they would be amenable to something like that. Right. Are there any other questions? If you think of any, feel free to send me an email or anything. I'm happy to answer any questions or connect you with some of the other folks who have been working hard on these CGC lists.
If it's any way useful to you, like years ago, I maintained a gene list database that had about 5,000 gene signatures in it. At the time we looked at, it was called GeneSigDB. We looked at a lot of approaches for doing that. And we did have like this issue with like HTNC symbols and the whole lot. So or possibly try to map to like ensemble or on-trade gene IDs or something else that was a bit more stable. But like I'm not maintaining it anymore. We still have all of those gene lists, but we did also work with our bioconductor at the time developing classes for gene lists and stuff like that. If any of it's useful for you, just let me know, and I'm happy to share the knowledge that we had that time. It was a lot of gene expression, gene signatures that we curated in cancer at the time.
Was there a specific cancer type that you focused on, or was it kind of all?
And cancer. It was 5,000 signatures. So
wow. Okay.
It was all manually curated from the literature at the time.
Oh, that's fantastic. Thank you so much.
So
I will definitely be reaching out.
Time. It was cancer, and we had some cancer immunology ones as well. But then kind of when everything moved into single cell, we stopped doing it. But it's all still on GitHub if any of it's useful.
Awesome. Thank you so much. I will definitely be reaching out to you.
Can you hear me?
Yes.
Okay. Yeah, we just wanted to chime in from the breast working group. Yeah, I think we kind of waded into gene expression, but only for those genes that are independently and individually clinically significant. But that is, definitely intriguing what you're saying, Eden, about gene expression signatures because we haven't quite made it into that. And for breast, it's highly important. Um, and then I guess
Yeah,
go ahead.
yeah, I started in breast. That was where the original impetus came from, was that we had developed a couple of signatures and we wanted to compare it to other breast cancer signatures at the time. So it started with whatever, about 30 breast cancer signatures, and it expanded considerably over many years.
Yeah. Um, yeah, I'm not really finding great resources as of yet for outside of proprietary databases and things for gene expression. And we really don't, have tests in the lab that we need to have an apparatus for interpreting them as of yet. Um, I guess the
Yeah.
other question I'll have is just more like mechanically. I think OB had made a comment too about hosting and just, having an online database. That is one thing that we also do struggle with is, online sharing. And so we're just using kind of a clunky, Google Sheets kind of mechanism, and even that comes with hurdles.
Mhm.
And it's not ideal. So having a web host site might be a good next step. So I like your comment, Beth, and CCGA might be a reasonable place to start with that. So yeah, and I
Mhm.
think also regarding maintaining the gene list to keep people engaged over the long haul, it takes a long time to get established and to, have a footprint where, people will go to that place to look up, a gene list, and then to have people stay engaged to maintain it is definitely been a challenge. Because yeah, the gene list will lose its relevance if it's not, continuously updated.
Right.
Yeah, when we did that years ago, we pulled a lot of those from the literature, and in each case we had kind of all of the different identifiers for the list, and then we would update it with each ensemble release. Um, because we wanted to work within Gen code where possible, because a lot of the gene lists had originally come from probe IDs in old microarray world. So rather than map to a gene, we wanted to map to where the probe ID location was. And that's kind of where we had lived within that world when we were doing it. But as I said, this was when we developed that a very long time ago. But we did have a whole suite of things where I suppose we also did a full tech search of every single PubMed paper and built a whole search engine for it. But then, yeah, we never maintained it after a while, because there wasn't really a lot of funding for it when single cell came along.
Right.
Yeah, if it's useful, we have all of the code. We have everything we did for that.
Well, thank you. We might reach out to you. I appreciate it.
Yeah.
Thanks everybody for the questions. I just dropped a link in the bottom too for those of you who are potentially interested. There's other ways to potentially map and understand like what you're explicitly representing. So the GA4GH RefGet sequence collections is designed so that you can potentially not worry as much about actually like the gene name, but if you know like the actual sequence that you are representing, you can do that and mint unique identifiers for being able to go back and actually like leverage those to help solve this as well. But there are a lot of efforts going on within the VICC proper and GA4GH for trying to work on harmonization and normalization, a lot of these different concepts.
Yeah, I forgot about that. I think that's might be a really good application for RefGet. Cool.
Awesome. Well, thank you so much, Beth, for your presentation. We really appreciate it.
You bet. Yeah. And if anybody has any questions or follow-up, just feel free to email me anytime. You can find me on Slack or wherever.
Awesome.
Thanks,
Thanks all.
everybody.